We can also think about doing it as one dimension. For instance, we can very easily take a string of beads that we bought somewhere and make an identical one simply by stringing beads together in exactly the same way. That would be replication in one dimension. Now, it turns out that in nature, this last paradigm is being used, namely the copying in one dimension. And you can see how this can be easily done, because both the RNA and proteins are essentially formed by linear one-dimensional arrangements of the building blocks. Now we come to the central dogma of molecular biology. The replication is done by DNA duplication. So DNA is the component where all the genetic material in the cell is stored. And if we want to make two cells, then we can make identical copies of the DNA. And now how do we make proteins according to the instructions by the genetic material? Well, this is a rather a complicated process because the DNA itself cannot be used to make proteins. We essentially need to copy the genetic information. And that's done by a process called transcription. The so-called messenger RNA is synthesized. Now, the messenger RNA simply represents an exact linear copy of the information that is on the DNA. And so at any given time, just a small part of the DNA is being transcribed into mRNA. And this mRNA then goes into the cell and finds itself a ribosome or several ribosomes. And then the ribosomes are used to make the protein exactly according to the linear instructions that are on the mRNA. Okay, so that's the central dogma of molecular biology. And so DNA contains the genetic blueprint. DNA DNA is used to make messenger RNA in a process called transcription, which also involve very elaborate molecular machines. And then I indicated there's a membrane here in eukaryotes, which is the nuclear membrane, so which in the mRNA goes through the nuclear membrane into the outer cytoplasm. And this mRNA then is used to make protein in a process called translation. So both transcription and translation are accomplished with the aid of molecular machines. And all this here we're going to ignore now, and we're going to concentrate on translation itself. Now, the person who is associated with the discovery of the ribosome is George Palade, and he found these intense granules lined up on the endoplasmic reticulum. So he says, note that in the matrix there are numerous small and dense granules, which he called G, which appear to have particular affinity for the membrane limiting the cavities of the endoplasmic reticulum. He didn't use the word ribosome because it didn't exist. It was coined only a couple of years later, and everybody got excited about this and was musing about what the nature of these granules were. Here you see a closer view of these. So what we know now about the composition of the bacterial ribosome is, and I'm talking about bacterial ribosome because there's a unity in life of these molecular processes. The ribosome is one of the most highly conserved molecular machine. And so when we study the ribosome of bacteria, then we know essentially how the ribosomes also work in multicellular organisms. So that's why I can just talk about the composition of the bacterial ribosome. So first of all, it is composed of two subunits, a small one and a large one. And each is now composed of RNA and proteins. So the RNA essentially is sort of the main skeleton, and many proteins are then sort of hung from it, or they partially interpenetrate the RNA. And the large subunit has many more proteins, and it also has two RNAs. And we talk about from the mass, we are talking about the 16S and the 23S RNA and the 5S RNA. And the proteins are called S1 through S21, L1 through L34, S referring to small subunit, L to large subunit. So the ribosome itself is multi-component, very, very complex, and then in itself it is composed of two subunits. The genetic code, this defines how a genetic sequence is translated into a sequence of amino acids. So the way it works is there's always a triple of bases, or for instance UGC or UGU is associated with one amino acid. There are 20 amino acids, 64 possibilities of drawing 3 bases out of a box of 4 bases. And so there are multiple assignments for certain amino acids. And then there's also a punctuation used, namely stop and start. Okay? So now the way it works here, this is a very simplistic diagram, but it shows you the main principle. The translation is accomplished with the help of a molecule which is called tRNA from transfer RNA. It has a recognition site which is called anticodon, and that anticodon recognizes the codon. And that recognition is really based on the recognition of bases. Okay, there's the CG, GC, AU are possible combinations of bases that fit together. Okay? Others would not fit. So this here is a so-called cognate fit because it will be recognized. Okay, if you replace this here with another one, with a G, for instance, then all of a sudden it would be a mismatch, and the whole thing would not fit. So the tRNA essentially arrives here and finds that there is a triple base codon, and then if there's a match, then it essentially gets stuck. That would be a simplistic way of expressing it. Then the tRNA gets stuck and presents its amino acid in the ribosome, so that the amino acid. I would appreciate if you just take, turn all these off, because it interrupts and it's very painful. So the next amino acid will then be attached with the help of the ribosome to the growth temperature. So this is all there is to it. So the ribosome has to make possible the arrangement of tRNAs on the ribosome, the tRNAs next to each other, and has to help in this whole recognition process. And the pairing, this correct pairing between the bases is called Watson-Crick pairing, because it was first discovered in the DNA. This is the basis for double-stranded DNA. The bases are exactly complementary.